I will start in like 3 minutes. Right, so I'll share my slides. So this is the third dive session where I'll first begin by discussing a doubt mentioned in the previous session. Then I'll discuss the lectures of week 3 and I will end with some practice problems. As always, the lecture and the slides will be uploaded. So if you're not attending the class, you can always see it later and send me questions, which I will address in the next class. So one doubt in the previous class was that there was this sample question that among these secondary structures, which is the DNA binding motif? So the answer was clear that it is the helix loop helix, where there are two helices connected by a turn. And one of the helices in the C terminal, just 1 second, I think someone wants to join. Just 1 second. Yeah, so what I was saying is that the answer was clear that it is the helix loop helix, which is the DNA binding motif. And in the helix loop helix, there are two helices connected by a turn. The C terminal helix is the recognition helix, which binds to the major groove of DNA, and the N terminal helix helps to position the recognition helix. So that was clear, but the question was asked that what is the SXXN motif? So I thought I'll also discuss the other three options, which are different secondary structures. So the beta alpha beta is quite simple. There is one beta strand, then an alpha helix, and another beta strand. And these two beta strands are parallel. So this is the beta alpha beta motif, which is pretty straightforward. Then you have the beta barrel, which are so you can, if we start from this beta strand, this is going down, the next beta strand is going up. So these two form an antiparallel beta sheet. And there are repeats of antiparallel beta sheets, and it is kind of, it forms a barrel-like structure. So several antiparallel beta sheets come together to form this kind of barrel-like structure, which has a hollow portion in the middle. So it can be used to carry some drug molecule or something which fits in the porous region in the middle. So this was also clear. The doubt was about the SXXN motif. So basically, S stands for serine, XX can be any amino acid out of the 20 amino acids, and N is asparagine. So when you have a tetrapeptide sequence in which the first amino acid is serine and the fourth amino acid is asparagine, it forms a SXXN motif. Basically, these motifs form a kind of turn structure. So you can see this helix here. At the end of this helix, there is a SXXN motif. So this region is um, uh, expanded and shown here. So again, I'll just see if, yeah. Yeah, so this region of the helix that you can see is expanded and shown here. This is a simple tetrapeptide sequence, but the uh, importance of this is that it acts as a helix breaker. You can see this is a helix that was there, and the SXXN motif is breaking the helix. So the helix ends here, and again, there are turn structures, and later a new helix forms. So wherever this sequence of tetrapeptide appears, um, there is a it is a break in the helix. So I'll read this part here. The SXXN motif in proteins indicates a propensity to disrupt alpha helical helices within the secondary structure, often forming a bend or turn. So you can see a kind of turn is formed by the SXXN motif. And this is because of the steric clashes caused by the bulky side chain of the serine residue. So serine has a CH2OH side chain, which is relatively bulky, and the polar nature of the asparagine residue. So serine and asparagine has a side chain of CH2CONH2. So both serine and asparagine have bulky side chains and polar side chains. So whenever they occur uh, um in first and fourth positions in a tetrapeptide, they act as a helix breaker, because if the, so we know that in helices there is interaction between the I to I plus 4. So this interaction will be broken up if you have serine and asparagine in first and fourth position, due to the bulky nature of the side chains, as well as the polar region. That is the SXXN motif. So this was a doubt asked in the previous class. I'm starting this class by first clearing this doubt. Now I will be discussing the different lectures in week 3. So week 3 was a basic introduction to crystals and X-ray crystallography. I'll start now. So in all living organisms, there are different levels of organization. As you know, all living and non-living things are made of one or more unique substances called elements, the smallest unit of which is the atom. These atoms combine to form molecules, which are the building blocks to all structures in the human body. So, the smallest level of organization is the atom, and atoms come together to form molecules. These molecules, when they are large in size, they are macromolecules. And we learned in the first class that there are four types of biological macromolecules: carbohydrates, proteins, nucleic acids, and lipids. Now, these macromolecules, the next level of organization after the macromolecule is the organelle. And organelles, as you know, can be of different types like ribosomes, um, Golgi body, and so on. And the different organelles come together to form cell. The cell is the smallest independent living thing in the human body. And the body is made up of many different cells, each with a particular function. But a cell is self-sustaining, meaning that that is why it's called the smallest independent living thing in the body. But an organelle, you can't call it an independent living thing, because the functions of different organelles work together to give life. Then in multicellular organisms like humans, the cells are organized into tissues. A tissue is a group of similar cells that work together to perform a specific function. So you can have skin, which is epithelial tissue. Then you can have your neurons, which form the nervous system tissue, and so on. The next level of organization after tissue is the organ. The organ is an identifiable structure of the body composed of two or more tissue types. Organs often perform a specific physiological function. Like kidneys are there for um, removing the waste in our blood. An organ system is a group of organs that work together to perform a specific function. So your excretory system is composed of the kidneys, ureters, bladder, and so on. So all these work together to excrete waste from the body. And finally, the highest level of organization, the most complex level of organization, is the organism. In this case, the human organism, which is composed of many organ systems that work together to perform the functions of an independent individual. So you can have the respiratory system, digestive system, excretory system, everything working together to form the highest level of organization, that is the human body. Now, in molecular biology, the most basic system is a two-component system, which is a basic stimulus response coupling or mechanism, allowing organisms to sense and respond to changes in many different environmental conditions. So suppose you touch something hot, the stimulus is the hot thing, and the response is that you move your hand away. So that is the basis of a two-component system. These two-component system consist of a membrane-bound histidine kinase. So you can see here the green is a histidine kinase that senses a specific environmental stimulus. So suppose the environment is giving you a signal that it is very hot. This signal is received by the histidine kinase, and a corresponding response regulator mediates the cellular response. So this signal will be converted to a response, and I will now discuss how that is done. First, the signal is transduced. So signal transduction occurs through the transfer of phosphoryl groups from ATP to a specific histidine residue in the histidine kinase. So you have ATP, which is the energy currency of the human body, and one phosphoryl group of ATP is transferred to histidine in the histidine kinase, and it becomes ADP. So now the histidine has a phosphoryl group. This is an autophosphorylation reaction. Subsequently, the histidine kinase catalyzes the transfer of the phosphate group on the phosphorylated histidine residue to an aspartic acid. Yeah, so I was discussing uh, the two component system in molecular biology, where the two component system is a basic stimulus response mechanism. That means, suppose there is a signal in the environment, suppose you're touching something hot, that is the signal, and the response is that you move your hand away. So that is the stimulus and response. So these two component systems consist of a membrane bound histidine kinase that is sensing the stimulus. After that, the signal is transferred so that the response can happen. And how this signal transfer happens, I'll discuss very briefly. So basically, you have ATP, that is the energy currency of the human body. And from ATP, one phosphoryl group or phosphate is transferred to histidine, and ATP becomes ADP. This is the autophosphorylation reaction. After this, the histidine kinase catalyzes the transfer of this phosphate group to an aspartic acid residue. You can see D is aspartic acid. So the phosphate group is transferred from H, that is histidine, to D, that is aspartic acid, and it is catalyzed by the histidine kinase enzyme. Now, this aspartic acid is attached to a response regulator or RR. And once the phosphate group is transferred, then the organism has a response. So basically, that is how the signal is received, and through transfer of phosphate groups, a response is triggered in the organism. Such kind of two component signal transduction systems enable bacteria to sense, response, and adapt to a wide range of environments, stressors, and growth conditions. Just 1 second, I think my yeah. So why is this important? That was the basis of communication. So signal and response is the basic of communication. Now, in biology, the communication happens through interactions. And in order to understand the process of communication, we need to visualize molecules in the atomic level and understand the three-dimensional arrangement of the atoms in the molecules. Now, there can be different levels of interactions, like there can be covalent bonds, hydrogen bonds, and even very weak van der Waals interactions. And also the size of the macromolecule can be very different. You can have proteins having only 50 amino acids or proteins having 2000 amino acids. So any system has complexity, and we have to identify the system to its lowest level of organization so that we can properly understand the 3D structure. So for that, a very high resolution is required, and for that, there are several techniques in order to picture a biological molecule at high resolution. And once we understand the molecule at high resolution, so suppose here you have two proteins interacting. So once you understand the structure of the molecule at high resolution, it will help you understand that what is the mechanism of this interaction, whether it is an electrostatic mechanism or there is some covalent bond happening. So once you understand the structure at the atomic level, the mechanism of action will become very clear, and that will help us in understanding the basis of diseases and developing new drugs. So there are several high resolution techniques in biology. Here I'll briefly discuss the ranges of the different experiments. So in the 20th century, a variety of experimental techniques were developed to examine the 3D structure of biological macromolecules. The most prominent techniques are X-ray crystallography, NMR or nuclear magnetic resonance, and electron microscopy. So you can see here, you can have eukaryotic cells which are in the range of 10 micrometer size, all the way to small molecules of the range of angstrom to 1 nanometer size. Now, if you're doing NMR spectroscopy, it can help you identify molecules ranging from 1 angstrom So NMR spectroscopy can help you identify molecular sizes from 1 angstrom to around 2-3 nanometer. X-ray crystallography has a slightly larger range up to around 10 nanometer. Single particle cryo-electron microscopy, it does not help in very small ranges, but you can see that protein and protein complexes are around the range of 10 nanometer. So proteins can be studied using cryo-electron microscopy. The other two we don't need to worry about right now, because in this course we will mostly be discussing NMR spectroscopy, X-ray crystallography, and cryo-electron microscopy. So basically in this class I'll be talking about X-ray crystallography. This had happened through the discovery of X-rays and the application to protein crystals. Once it was found that X-rays can be used to determine the three-dimensional structure of protein crystals, structural biology was revolutionized, because now scientists could obtain the three-dimensional structure in atomic detail. Similarly, NMR will also give you the three-dimensional structure, but additionally, NMR will also give you an idea of the dynamics of the molecule. So in X-ray crystallography, you are basically fixing the molecule in a crystal. So it won't give you an idea of the dynamics, but NMR will also give you an idea of the how the molecule is moving. And the latest technique in the 21st century is the cryo-electron microscopy. Basically, what this does is that in X-ray or NMR, there can be certain disadvantages. Like in NMR, high concentration is required. In X-ray, you have to form a good crystal, otherwise the structure will not be well resolved. So cryo-electron microscopy takes care of all these problems, and it is mostly like through reconstruction software that cryo-electron microscopy gives the highest resolution in protein structure. Of course, the disadvantage is that cryo-electron microscopy cannot resolve beyond around, say, 5 nanometer. So that very small proteins you cannot resolve by cryo-electron microscopy. So you can see here the ranges are given. X-ray crystallography has range from 1 to 3 angstrom, NMR around 2 to 3 angstrom, and cryo-electron microscopy greater ranges from 3 to 4 angstrom. In this class, mostly we will be discussing the X-ray crystallography. So these are some basic terms that are used when studying waves. So in a wave, an amplitude is the distance from the equilibrium position to the top of the crest or the bottom of the trough, the vertical distance basically. So this is the amplitude. Also, there is something called a phase. Phase is the relationship between two or more signals that share the same frequency. So here are two waves having the same frequency. The red wave, if it is moved by 90 degrees, it will overlap with the blue wave. So the phase between the red and blue wave is 90 degrees. You can see on x-axis the degrees are plotted. So just imagine that you are shifting the red wave towards this side by 90 degrees. Then it will completely overlap with the blue wave. So it is said that the phase relationship between the two waves is 90 degrees. Now there is one problem in X-ray crystallography, that whenever you have a diffraction pattern, so basically the X-rays are passed through the crystals, and then the X-rays are deflected by the electrons and atoms in the crystals, and you get a pattern called the diffraction pattern, which looks like this. From the diffraction pattern, you will get an idea of the intensity, that is the amplitude of the X-ray. However, you will not get any information about the phase. So the phase information is completely lost, and that is called the phase problem. But amplitude and phase are both very essential in order to reconstruct the three-dimensional structure of the molecule. So here we are just defining the problem. How the problem is solved, we'll discuss in the next class. So this is the entire electromagnetic spectrum. Ranging from radio waves, which have the largest wavelengths, 10 to the power 3 to 10 to the power minus 1 meters, all the way to gamma rays which have the smallest wavelength. And you can see that in each wavelength range, what are the different degrees of organization. So X-rays can detect atoms because you can see the size of atom, it falls within the range of the X-ray, that is 10 to the power minus 8 to 10 to the power minus 11 meters. So in order for an object to be visible under magnification, the wavelength of the light that is given here must be, roughly speaking, no larger than the object. So the problem is that visible light, you can see visible light here is much greater than the dimensions of the atom. So the dimensions of the atom are quite small. They are in this range of 10 to the power minus 8 to 10 to the power minus 11 meter. But visible light has much greater wavelength. 10 to the power 15 to 10 to the power, 10 to the power minus 4 to 10 to the power minus 7 meters. That is why visible light cannot be used to resolve up to the atomic level. And so X-ray is the only one that can be used because it is within the range of the atoms. So now there are some basic questions that we need to address. Why can't we visualize the molecule directly with the X-ray? Why do we need to form a crystal? Basically, a single molecule is a very weak scatterer of X-ray. Most of the X-ray will pass through the molecule without being detected. So if you have a single molecule and you put an X-ray on it, it will just pass through the molecule. There will be no diffraction and no pattern will be formed. So in order to solve this, the molecule is crystallized. Basically, a crystal is a three-dimensional repeat of ordered molecules whose signals reinforce each other. So instead of a single molecule, you have around 10 to the power 14 molecules in an ordered arrangement. So when there are so many molecules and you pass the X-ray through the crystal, now there is sufficient electron density to diffract the X-rays properly and create a strong diffraction pattern. And because of the regular arrangement in the crystal, so suppose the X-ray diffracted from this molecule and the X-ray diffracted from the neighboring molecule, they will reinforce each other, meaning they will be in the same phase as each other. And so the resultant intensity of the X-ray will be increased. So very strong diffraction patterns will be formed from a crystal compared to the molecule which has a very weak diffraction pattern. That is why in X-ray crystallography, the first step is to crystallize the molecule. The second problem is that,
Excuse me, ma'am.
yes.
Ma'am, how always we are saying that uh, the molecules are 10 to the power 14, means in every crystal we will get 10
No,
to the
no, no,
power
no. That
14.
is just a average size. Like you can have less also, you can have more also. So basically, what is done is that you first crystallize the protein. Uh, then you see whether the crystal is forming a good diffraction pattern. If it is forming a good diffraction pattern, it is fine. If it is not, that means you need a larger crystal. Then the conditions of crystallization is changed in such a way that a larger crystal is formed, and so you get a better diffraction pattern. So it is kind of a trial and error thing where depending on the size of the individual protein molecule, the crystal size will be modified.
So, ma'am, this thing takes a lot of time because it's trial and error. So, um,
Yeah, protein crystallization is a very difficult process. That is the main drawback of X-ray crystallography, is that, so you will see any lab which works on crystallizing a protein, it will take them almost 1 year to get proper, like uh, well resolved crystal structures of the protein. So that is actually why now NMR and cryo-electron microscopy is used more compared to X-ray crystallography.
ma'am, and in the last, means in the previous slide, you were showing us that two, no, ma'am, more
This slide or
previous.
the one
No,
before
ma'am.
this?
Yes, ma'am, the one before this.
Haha.
Ma'am, in this, what you were saying, actually, my network problem was there
Oh,
at that
oh,
time.
I was just defining the terms that amplitude is from the uh, peak to the equilibrium position, and phase is the, so suppose you have two waves, then the difference between the so how much you have to move one wave to superimpose with the other wave, that is called the difference in phase between the two waves. So this red and blue wave, the difference in phase is 90 degrees. So these are two terms with which in next class we will use these terms in mathematical equations to see different things. So here I'm just defining the terms. And one problem I was saying is that whenever you do X-ray crystallography and you get a diffraction pattern, the diffraction pattern will basically only give you the intensity, that is the amplitude squared. So from the diffraction pattern you can get the amplitude of the wave, but the
Mm.
phase information is completely lost. So this is a problem, and how we deal with this problem will be discussed in the next class. So I was just introducing these terms.
Okay, ma'am. So you started with that response regulator means the type 3 component system.
Yeah.
The first topic of this presentation was type 3 component system.
Before that, there was just a basic slide of different levels of organization, like from atom, you can go to molecule and so on. So this was just an introduction to the different levels of organization.
Ma'am, like sir has taught in that video, the same way you are going.
Huh.
Okay, ma'am.
This basically, the live sessions, I basically cover whatever was taught in week 3. So this is a third live session, so I'll cover whatever is taught in week 3, and I will discuss some sample problems. So that is, and you can clear
Okay,
any
ma'am.
doubts. So that is how the live
Yes.
sessions will go.
Okay.
Yeah. So the next thing is that unlike visible light, X-ray cannot be focused using lens. So in visible light, you can have a lens and there are light rays, and after passing through the lens, the light rays will all be focused at a focal point. That can help you magnify or uh, reduce the image of something. That is how a normal light microscope works. But why an X-ray cannot be used like this? Because when an X-ray passes through a lens, the refractive index is 1. So visible light, when it passes through a lens, it will bend. But X-ray, it will just pass through straight. The refractive index 1 means that this angle is the same in the incidence and refraction. So if an X-ray is put, it will just pass through straight. So using for an X-ray, you cannot use a lens. The solution of this is that a computer is used in X-ray crystallography. Always there is a computer that is there which simulates the image reconstructing lens. That means you don't use normal glass as lens, but the computer interprets the diffraction pattern and acts as a lens, and then displays the three-dimensional structure on the screen. So you in X-ray crystallography, the normal lens used in light microscopes is not used. So now,
Ma'am, why
huh,
the X-rays pass in a straight line?
um, actually, like the refractive index, which is basically what causes the bending of light rays, is the speed of light in air and a material. So normally in normal materials, light gets slowed down because the wavelength of light is big enough that when
Yes,
it
ma'am.
enters but X-ray has very small wavelength, so it just passes through the gaps in the medium.
Okay, ma'am. Mhm.
Okay. Now we'll just talk about the different crystals. So an ideal crystal is a perfect crystal where each atom has identical surroundings. You can see this first one is an example of an ideal crystal. But any real crystal has a large number of defects or imperfections. So here are some examples of defects. Like you can have an interstitial impurity where there is an additional impurity in the gap of the crystals. Or you can have a cation or anion vacancy where one of the spots are empty. or you can have a substitution of cation. So some other cation has come and sat in the place of this. So suppose instead of Na plus, you have K plus. So that is a, or the opposite, like the anion has also been substituted. So these are different defects in the crystals. And these, more the defect is there, the worse your X-ray diffraction data will be. So the first challenge is obtaining a perfect crystal. Now, why real crystals are not perfect is because a perfect crystal is of infinite size, but real crystals are of finite size, and so they have a surface boundary. And at the boundary, the bonds are terminating, so there is an imperfection created on the boundary. So you can reduce crystal defects, but any real crystal will have some level of defect, and that will be later corrected by different software. That also we'll discuss later. Now, there is a difference between salt crystals and protein crystals. One major difference is that protein crystals has a large amount of solvent. So protein crystals, the solvent percent is almost 30 to 80% solvent. So whereas in salt crystals, if you have studied NaCl crystals in inorganic chemistry, you can see there is no solvent at all. It is just alternating Na plus Cl minus. So because of the large amount of solvent in protein crystals, a very small fraction of the protein surface is involved in crystal contacts, and the rest is exposed to the water that is inside the crystal. So because of this large water content, protein crystals are very soft and easy to crush compared to salt crystals. Another thing is because of the presence of solvent inside the crystal, small conformational changes can happen within the protein crystal without damaging the crystal. And because these conformational changes can happen, a protein in a crystal form can also act as enzyme. So how the enzyme acts is basically it has an active site, and the substrate comes and attaches to the active site, and there is a reaction that happens which changes the substrate into the product. Now, even if the protein is in crystal form, because of the presence of a large amount of solvent inside the crystal, the protein can undergo the necessary conformational changes required to behave as an enzyme. So even in crystal form, the protein can behave as an enzyme. There are certain problems that happen in protein X-ray crystallography. Again, here just the problem is being introduced. We'll deal with it later. Is that one thing is birefringence. This is a sorry. This happens in salt crystals, because salt crystals are determined using um, visible light, and so because protein crystals are determined using X-ray crystallography, there is no such defect, because again, as I said, that in protein crystals, the X-rays will just pass through the crystal without any um, deviation, whereas in salt crystals sometimes you can have a defect called birefringence, where you have the visible light uh, um, deviating in different angles at different surfaces. So the final image, suppose the image was this big, after passing through the light, the image kind of becomes distorted. So that is a problem that happens when you have optical lens or you're using visible light. So this problem is precluded in excluded in your protein X-ray crystallography. So again, these are just some facts about the crystallography process. Now, one thing is
Um,
that, huh,
so if in the what in the visible light, if that
mhm,
light is passing through the object,
mhm.
then it is good that means it is not good that it is there is something change in that particular object, means image of that particular object. But ma'am, in X-ray crystallography, the same is being formed as a image.
No, in X-ray crystallography, you are not using light waves, no? So, and I discussed that the refractive index of the X-ray is 1, so it will just pass through directly. So, this problem arises only when you are using visible light and you are using some optical material.
And then what is the problem with X-ray crystallography?
No, there is no problem. I am just like,
Okay.
this was just a point that sir discussed. That is why I'm mentioning it. This has
Mhm.
nothing to do with X-ray crystallography.
Okay, ma'am.
Okay. Now, there is a topic was discussed, which was basically cloning DNA using vectors. So, mostly the organism used for this is E. coli, and that is because there is no ethical concern about growing, manipulating, and killing E. coli. And also, the E. coli can reproduce very rapidly. So, the experiment time becomes very fast. So, every 20 minutes you can have a new population of E. coli. So that is another reason why it is used, and also because it can survive in a variety of conditions. So the first and plasmid is used in order to clone the DNA. So basically, in the plasmid, the gene or DNA of interest is inserted after a promoter region, and you can add certain um, uh, genes called selectable markers. So suppose you want the plasmid to have antibiotic resistance, so certain characteristics like antibiotic resistance can be inserted by inserting the required gene. So once the plasmid with the DNA of interest is inserted into E. coli, it is allowed. So you can see this is the plasmid, one part of the plasmid which is non-coding DNA, that is, it is not DNA which codes for anything important, that is cut off with restriction enzymes, and the target DNA or gene that you want to replicate is attached with DNA ligase to form the recombinant plasmid. Then the recombinant plasmid is introduced into the E. coli bacteria, and there is some selectable marker that in red here, which helps us identify which plasmid has been modified and which has not been modified. And then the E. coli bacteria is allowed to grow and reproduce. Then in your culture plate, you will have some E. coli bacteria which has the recombinant plasmid, which is shown in white here, and some E. coli bacteria which does not have, which is like not affected, which is shown in blue here. So you can choose which E. coli bacteria has the gene of interest, and that will help you get multiple copies of the same gene. So why this DNA cloning is done is because sometimes in some experiments, a lot of copies of DNA is required, so it is easy to copy the DNA using this recombinant DNA technique or DNA cloning. So the bacteria or E. coli is used as factories in order to replicate this DNA. So now we'll discuss some techniques of protein purification. So one
Ma'am,
technique, yes.
ma'am, so one, I have a doubt. Ma'am,
Mhm.
when this, one second, ma'am, actually, ma'am, in the diagram means how we will know that it is the non-coding region?
Like the region that is cut off, you are saying, right?
Yes, ma'am. How we will know that
So
this particular region is not means is not required for the bacteria, means is not useful for the bacteria, and so we'll cut that portion.
so basically, in the first class, I had discussed the there are some techniques of DNA sequencing, right? And by DNA sequencing, you can identify that which are the sequences of DNA that are participating in forming important proteins, and which are the sequences of DNA which are not active. So these have already been determined by biologists, and there is a library. So there is a protein data bank. There is a nucleic acid and DNA sequence data bank also, which is
Yes,
kind of
ma'am.
like a library. So using that library, it is now being done.
And after inserting that the particular desired DNA, how we will know that with the help of ORIC site that this DNA is being inserted in the mm plasmid. That means the desired DNA is inserted in the plasmid, because previously also the ORIC site was there, and now also means, sorry, not ORIC site, the marker gene. Previously
Huh,
also
huh.
the marker gene was there, and after inserting the sequence, desired sequence, now also marker gene is there. Then
Okay.
how we will know that?
That is actually a good question. I don't know. I'll find out and I'll let you know in the next class.
Okay, ma'am.
Okay. So now we will be discussing some techniques of protein purification. Firstly, there is the gel filtration chromatography, where the proteins are separated by size. So in this, there is a column and there are beads in the column, and the beads have certain pore size. So here you are adding a mixture of large protein and small protein. So the red ones are the small proteins, the blue ones are the large proteins. Now the beads are chosen in such a way that the small proteins can get inserted into the beads, but the large proteins will elute out. So now when solvent is passed through the column, the large proteins are loose and they will leave the column first. So you will get the large protein first, and then eventually when you keep adding solvent, the small proteins are obtained after a longer time because they were absorbed onto the surface of the beads, and so it will take more concentration of solvent to remove the small protein later. So here the entire concept is based on the size of the protein, and the beads that you are choosing, they will have to have appropriate size such that only one protein will be absorbed on the bead and the rest will pass through. So for this also you need to have a good knowledge of the pore size in the beads and the protein size and so on. And the other technique is the ion exchange chromatography, where the separation is based on charge. So here in the column, there are positive. Are you saying something? Sorry, I heard. So in the column, you can either have negatively charged columns or you can have positively charged columns. In this example, it is negatively charged columns are there. So when you are adding proteins, uh, firstly the positive, suppose you have are adding a mixture of positive and negative proteins. By positive proteins I mean proteins which have more basic amino acids. You know, if there is basic amino acid, there is positive charge. And by negative proteins I mean proteins which have more acidic amino acids, so the net charge will be negative. So suppose the surface of the column is negatively charged. In that case, the positively charged proteins will bind to the surface and the negatively charged proteins will get repelled out first. Or if it is a more complicated case, like it is shown here, you have both positively charged proteins. So both the proteins are getting attached to the surface first. Then what is done? A solution of NaCl is added. So suppose the protein which has lower affinity for the column, that will be removed first. So NaCl is coming and replacing the protein. So the protein is being released from the column. And the protein which has higher affinity for the negative charge, because they have higher charge, they will be stuck in the column. And to remove that, you have to use even higher concentration of NaCl. So basically, the way this separation works is that if the protein has lower charge, you can remove the protein by adding low concentration of NaCl. And if the protein has higher charge, you have to remove the protein from the column by adding higher concentration of NaCl. So by gradually increasing the NaCl concentration, the proteins can be separated And this separation technique will only work when the proteins have sufficient difference in their charges. Another kind of this separation is called affinity chromatography, and this is based on very specific biological interactions between two molecules, such as interaction between enzyme substrate, receptor ligand, or antibody antigen. One example is the nickel affinity chromatography. So the column contains a compound of nickel. So in the column, we don't need to go into the details of this, but there is some compound that contains nickel. And if you are adding a protein which has histidine on it, so that protein will specifically bind to the nickel and remain in the column, whereas the protein which does not have the histidine will pass through and be separated. And in order to remove the protein that is stuck to the column, imidazole imidazole is added because imidazole has a higher binding affinity with the nickel compared to the histidine. So then the imidazole will replace the histidine and the protein will be eluted out. So this type of column is based on very specific interactions, like this one is interaction between nickel and histidine. Then you can have other interactions also. This is the one we are discussing right now. So this type of column is very specific to certain proteins. You can't use them generally for all proteins like the size exclusion or the ion exchange. So this is the concept of affinity chromatography.
Means imidazole will have higher affinity with nickel than histidine.
Correct. So it will
Ma'am,
preferentially bind with nickel, and the protein with the histidine will be removed and can be collected.
ma'am, protein with histidine or just only histidine?
No, see, this thing is about separation of proteins. So this blue
Okay.
one is a protein, and this orange thing that you can see is the histidine attached to the protein. So
Okay,
any
ma'am.
protein having histidine will get attached to the nickel tag, and the protein which does not have histidine will be removed from the column. So you can collect it separately. Then if you add imidazole, imidazole will come and attach to the nickel, and you can separately collect the other protein which has histidine. So then the two proteins can be separated.
Ma'am, and in the case of ion exchange chromatography, the separation is based on the charge,
Yes.
means whether it is cation. Ma'am, so suppose means if a protein is having basic and acidic, means it is having more basic and less acidic.
Huh.
Uh,
So positively charged.
yes, ma'am, positively charged. So the matrix will be of negatively charged ions.
Correct. Correct.
And ma'am, ma'am, and suppose ma'am, one I have a doubt. If
Mm-hmm.
uh, if we are placing a multiple of proteins, means multiple types of proteins to
Mm-hmm.
separate on the basis of charge,
Hmm.
and in that case we have two types of protein, one which has more basic charge and one which has more acidic charge, means one which is negative and one which
Right.
is
Right.
positive.
Huh.
So
Huh.
ma'am, the matrix is of negative charge.
Huh.
So
So, hmm.
in that case what will happen?
Huh. So if you add both negative and positive proteins and the matrix is negatively charged, it will
Hmm.
repel the negatively charged protein. So the negatively charged protein will be eluted out very fast. Like if you just add the positive and negative proteins in one wash of the column, the negatively charged protein will be washed out because the column will repel the protein. So it will not stick to the column at all. So you just have to pour it with the solvent once and the negatively charged protein will come out. So that separation is actually much easier compared to if you are separating two positively charged proteins.
This means one more case is possible also. Suppose means I am saying that the matrix is of negatively charged, and
Mm-hmm.
the protein is of positively charged. Then negative will attract positive, and it will form, means positive will bind positive proteins will bind to means basic proteins. Now basic proteins will
Ha ha.
bind to that matrix, and
Correct.
the acidic proteins, they can also bind to that basic proteins. Now means they can also bind to that basic proteins, because positive will attract again negative, and like this way they can form a complex. Is this possible?
I guess that is also possible because I guess it depends on the whether the interaction. So you are first dissolving both the proteins in a solvent, right? So it depends
Mm.
if the protein-protein interaction is larger or the protein-solvent interaction is larger. So definitely, if the protein-protein interaction is larger, this kind of thing can happen. That is one of the reasons this ion exchange chromatography often there is blockage in the column. So when protein protein aggregates form, there will be blockage in the column. So then either the solvent has to be changed or the this matrix has to be changed. So that is one disadvantage. You are correct, that can definitely happen if the protein protein interaction is very strong.
Yes, ma'am. Okay, ma'am.
Yeah. So now I will be discussing some techniques of crystal formation. So the most common technique is called vapor diffusion. In this method, what happens? Uh, there is a solution like this, and from the solution, evaporation is happening and droplets are forming, and there is an equilibrium between the solution and this droplet. Now, this droplet contains the purified protein, a buffer solution, and a precipitant which is helping the protein to precipitate. Now this equilibrium is allowed to happen. So both the this is called the reservoir and this is the droplet. So both the droplet and the reservoir has a solution of the protein, the buffer, and the precipitant. Initially, as the drop is forming, the protein concentration is low, but as there is equilibrium, the protein concentration in the drop gradually increases, and if you use the appropriate crystallization solution, protein crystals can start to form in this drop. So this is one of the methods of forming of protein crystals. And advantage is that because the equilibrium is a very slow process, it allows for gentle and gradual changes in concentration of protein and precipitant. So that helps in formation of large and well ordered crystals. So this kind of vapor diffusion can be done in two ways. One is the hanging drop, which is the one which is shown here, or another is the sitting drop format. So sitting drop is similar concept, but instead of hanging from the drop, the hanging from the top, the drop is forming at the side. Similar, both these methods are based on the concept that there will be a equilibration uh, and slowly the concentration of the protein in the drop. So initially a high concentration of protein in the solution is taken. So when the equilibration happens, slowly the concentration of protein in the drop will increase. And if you take solvents that support crystallization, as the concentration increases, the crystal will begin to form. Another method is microdialysis, where you can see here the solution of protein, buffer, precipitant, everything is taken in a semi-permeable membrane. And across this semi-permeable membrane, small molecules and ions can pass, but proteins cannot pass. So, uh, in the again here there is a concentration that is, so here it is the opposite thing. In the first two methods, the reservoir had a large concentration and the concentration in the drop was gradually increasing. Here you take a large concentration inside the semi permeable membrane, and gradually you allow the solvent and the precipitant to enter the semi-permeable membrane from the reservoir. Now, when the appropriate concentration is reached, crystallization will start to happen. So, basically, crystallization, I'll discuss in one of the coming slides. Crystallization happens at a very specific concentration. And if we suddenly reach that concentration very fast, it will be like the crystal ordering will be very bad. So the crystal structure will be imperfect. So the goal is to gradually and very slowly either increase or decrease the concentration till we reach the optimum concentration for formation of crystals. That is why this kind of equilibrium or semi-permeable membrane also slows down the process, because the exchange through semi-permeable membrane is very slow. So that is also slowing down the process. So gradually you reach the optimum concentration, which will allow the crystals to form. So this is the graph I was talking about, is that there are several zones, it is called. So this line, the dark blue line, is called the solubility line, and it divides it into two parts. This entire white region is where so there is a protein concentration, there is a precipitant concentration. Precipitant is basically a molecule that helps in crystallization. It is not part of the crystal, but it helps to bring the protein molecules close together so that they can form crystals. So there has to be a very uh, balance. Has to be a very good balance between the concentration of protein and precipitant. Suppose the protein concentration is in this white region, but the precipitant concentration, so in this white region, it is basically called under saturation. That means the protein molecules will stay in the solution. They will not form crystals, they will just remain in the solution. So this is called the soluble state, in which the protein molecules are soluble. Then as you move towards the right, it enters the supersaturated region. So this light blue and dark blue, they form the supersaturated region. And the difference is that in this light blue region, basically the crystal growth happens slowly and gradually. But in this dark blue region, nucleation happens. That means, so sometimes, like when you want crystals to form, suppose copper sulfate, you have a solution of concentrated copper sulfate, but crystals are not forming. So you add a very tiny crystal of copper sulfate in the solution, and around that tiny crystal, very rapidly large crystals will begin to form. So that is called a kind of seed crystal, around which the larger crystals will begin to form. That is what happens in the nucleation zone, is that the seed crystal or first crystal starts to form, and then immediately as the nucleation happens, the crystal will begin to grow. So I'll just, in the crystal growth zone, that is this light blue region, only transient nuclei are formed that do not reach the critical size. So in crystal growth zone, there is a kind of equilibrium happening. So in crystal growth zone, there is an equilibrium happening. Crystals are forming, then they are again breaking and becoming soluble. They are not forming proper large crystals. But the moment you enter the nucleation zone, the critical size of the crystal is reached. And so from that nucleus or seed crystal, large crystals can begin to form. So you can see, if the supersaturation proceeds, the nucleation zone is reached, where nuclei can achieve a critical size and become stable. Then you can get stable formation of large crystals. Now, as the nuclei is becoming larger and crystals begin to appear, the protein concentration in the solution decreases. So this equilibrium that is there, that equilibrium will again get shifted, because when the crystal starts forming, protein concentration decreases, so it will again go back into the soluble state. That is why this equilibrium is very essential, because when otherwise crystals will start to form and they will stop. But in this case, what will happen? Crystals will form, and then so the concentration in the solution decreases, so more protein will come up and condense here, so that the concentration can be maintained, so that the crystal can continue to grow. And
Ma'am, not getting, ma'am, this thing.
see, slowly you are increasing the concentration of protein from left to right. Okay, so when you are reaching the nucleation zone, a nucleus is forming, which is basically a crystal of which crosses the critical size. And when it crosses the critical size, the crystal can continue to grow. So then the protein in the solution, the concentration of the protein in the solution will become less because the protein is crystallizing out, right? So in order to keep the protein concentration in solution high, more protein from the reservoir will evaporate and come here so that the protein concentration of the solution remains high, so that the crystal can continue to grow. Otherwise, so when you are in this concentration, crystals will form. But when the crystal is forming, protein is getting removed from the solution. So crystal should stop after reaching a certain point, because it will again go back to this soluble state where the protein concentration was low. So that this does not happen, you have an equilibrium with a reservoir which has a large concentration of protein. So whenever the solution protein in the solution concentration is becoming low, more protein can come from the reservoir into the solution so that the crystal can continue to grow.
Ma'am, can you explain the last sentence? I'm not getting.
So see, crystals are forming, right? It is in the nucleation zone and crystals have started to form. So because whatever protein was there in the solution, it is crystallizing out. Protein concentration in the solution is low. Now more protein can, so what is this equilibrium? The equilibrium maintains the protein concentration of the reservoir and the drop to be the same. But if protein in the drop is crystallizing out, then the concentration in the drop becomes low. So again, more protein will come from the reservoir and condense in the drop, so that the protein concentration is maintained at the concentration required for crystallization.
Concentration in the drop starting in the starting protein concentration will be low in the drop. From the
Yes,
reservoir, it will go in the that drop to
correct.
crystallize the protein.
Ha,
Then after a certain time, they both will maintain equilibrium, because if they will not maintain equilibrium, then from the drop the proteins will again get evaporated to the solution and the crystallization will not be formed.
correct. Ha.
But after that, what will happen?
So
This is
once the concentration in the solution decreases, you have to add more protein into the solution from outside. So the
okay.
concentration, so when crystals start forming, automatically the concentration in the drop will decrease. In order to increase the concentration in the drop, the reservoir will supply more protein. So protein
Okay.
concentration in the reservoir will decrease. So you have to add more protein from outside in order to uh, balance it out. Only then
Okay.
more and more protein will be able to come to the drop and get crystallized.
Okay. Means the basic concept is this only.
Yes, yes. And then if you saturate the solution too much, then you reach the red zone, which is the precipitation zone. At that time, crystals don't form, but the protein will just aggregate and precipitate out. So you want to maintain the concentration within the nucleation zone. So then the concentration of the reservoir has to be adjusted in such a way that the concentration in the drop is maintained in the nucleation zone, because it is in this zone that you have the best formation of crystals. I'll proceed.
Okay, ma'am.
So there are some challenges involved in protein crystallization. One is that if the saturation is too much and too many nuclei form. So suppose you have one nucleus and that is growing into one big ordered crystal. That is ideal for X-ray crystallography. But suppose you have many tiny nucleus forming. Then I discussed before also that size of nucleus is an important factor. So this is not a very ideal situation. So sometimes that is one problem that happens. Another problem that happens is that sometimes nucleation does not happen spontaneously. So you have to add some crystal from outside of the same protein, which is the seed crystal. And around that crystal, the rest of the crystal will begin to grow. So these are some challenges that is faced during protein crystallization. And here I've just defined the unit cell, which, as you know, is the smallest repeating unit in a crystal. Uh, we don't need to go into the details right now, but the unit cell is the basic building block of a crystal. So there is an initiative by Protein Structure, initiative of the NIH USA, which determines the three-dimensional structure of biologically relevant proteins. And it aims to, so whatever three-dimensional structure it determines, it puts in the public domain so that people can research on it without any problems. So in the first class, I discussed the protein data bank. So in the protein data bank, these protein structures are deposited so that any researchers from all over the world can access these structures and study the different interactions in order to design drugs and so on. So there are different goals of this initiative. Some of which are like purifying proteins and crystallizing proteins in order to study them by crystallography, determining the 3D structure of proteins, then seeing like suppose you have a family of proteins which have one common region. That means that common region has some function. So there are branches of biology which focus on determining the function of specific folds of proteins based on homology modeling. Homology modeling is finding out the common region of protein in several organisms and so on. So these are different goals of that project. And that is why it's very important to find out the 3D structure of the protein. And this is some basics of X-ray crystallography. This I'll go into more detail in the next class, but I'll just discuss it shortly here. So you have two, so this is a crystal that is shown here. So this blue dots are the single molecules, and this is a crystal that is shown. So you can have multiple, one X-ray will come. So suppose you have one molecule here. The first X-ray is bouncing off the molecule on the first layer and it is reaching here. The second X-ray is passing through the first layer and bouncing off a molecule in the second layer of the crystal and reaching. So both the X-rays, when they reach the collector region, if they are both in the same phase, there will be constructive interference. You can see the lower figure shows constructive interference. So this is also in positive, this is also positive. So there will be constructive interference, that is the amplitude will be larger. And this is an example of destructive interference. One wave is positive, one wave is negative. So this will completely cancel out. So like this, different X-rays will diffract off different layers of the molecule of the crystal, and you will get a net. We always prefer a net pattern that is constructive, because that is the entire point of having crystals where you have. So we studied earlier that what the advantage of a crystal is, that there are many molecules in the crystal. So one X-ray deviated of each molecule, and if you manage the wavelength and everything in such a way that these X-rays will, the diffracted X-ray will be constructively interfering, then you will get a nice high intensity diffraction pattern. So we'll go into the mathematics and everything later, but that is the how the parameters are adjusted so that so say the distance between the crystal layers, or the wavelength of the incident angle at which the X-ray has been shined, all these are adjusted in such a way that the net interference that happens is constructive interference, that all the waves are in the same phase. So you can see that if you get a perfect crystal, you will get very well defined dots in the diffraction pattern. And if there are some defects in the crystal. Rotational disorder means that, again, we'll discuss these later, but there are certain disorders called rotational disorder or translational disorder, and you can see the dots are becoming blurry. So why that is happening is only when there is a periodic distribution, you will get such perfect constructive interference. If there is any disorder, so the molecule is not where it should be in a crystal, then there will be destructive interference and the intensity of the net X-ray will be reduced. That is why you will get blurry patterns like this. Again, in this class, I'm discussing in a very qualitative way just to introduce you to the different topics. Why this happens, how this happens, we'll all go into that later. Okay, now for the energetics of what is happening in crystallization. So you have a protein in solution and that is in equilibrium with your protein crystal. Now you know that delta G is delta H minus T delta S. Now the delta H of crystal formation is modestly favorable from 0 to minus 17 kilocalorie per mole. So we know delta H being negative is favorable, and it is said to be modestly because it is not that high of a number. For enthalpy, like enthalpy for covalent bonds is much higher. So this enthalpy is small, so it is said to be modestly favorable. So this is pushing the equilibrium towards the right. Now, in the delta S, you have two different delta S. One is the entropy change of the protein, another is the entropy change of the solution. So imagine you have two protein molecules shown here by the blue bars, and they are separately solvated. When crystallization is happening, the protein is coming together and all the solvent molecules are released. So the delta S protein is unfavorable. Entropy is decreasing, right? Because earlier there were two protein molecules and now there is one aggregate. So the entropy of the protein is decreasing. So delta S is negative. So, delta S is negative, and so minus T delta S is positive. So the T delta S protein term is unfavorable because the entropy of the protein is decreasing. And the T delta S solvent is favorable because solvent earlier it was trapped between the protein molecules. Now the solvent is free. So the entropy of the solvent is increasing. So the T delta S term is positive and there is a negative term outside. So this becomes a negative quantity. So the delta H crystal term is negative because it is slightly favorable enthalpically and it is a small term. Delta S protein, it is decreasing. So minus T delta S is positive and delta S solvent is increasing. So minus T delta S is negative. Overall, the second two terms more or less cancel out and you get the Gibbs free energy change of formation of the crystal as a very small negative value. So as long as the delta G is negative, the process will be favored. Spontaneously, it will occur. But you have to remember that the delta G is still small. So it is not like it is a very fast process. That is why I was saying that protein crystallization is a very slow process because the net magnitude of delta G is still small. But overall, thermodynamically, it is modestly favored the crystal formation. Are there any questions here? I'll just move on. You can ask later. So how do we increase the how do we make the process of crystallization more favorable? One thing is that to reduce, so the delta S protein is unfavorable because initially the proteins are all free and then they are forming the crystal. So the entropy is unfavorable. So how can this be reduced? The unfavorable entropy change, how can we reduce it? One way is to cut out. So if the protein has a long hanging chain, you can cut it off. Uh, Basically, the goal is to reduce the entropy of the protein in solution. That you can do by removing any long hanging tail, or uh, you can crystallize. So suppose there are regions of the protein that are independently moving around. If you add a ligand or something to lock the protein in place, then the entropy of the protein in solution will decrease. And so the net entropy loss that is happening, that will also decrease. Or suppose you have high entropy residues like glutamic acid or lysine, which have long side chains. Long side chains mean that the entropy is high. So we can mutate those residues to alanine, cysteine, or serine, which have short side chains. So when there are short side chains, the initial entropy is low. So the net loss in entropy on crystallization is also reduced. So some modifications in the protein like this will help us reduce the loss in entropy that is happening when a protein is going from solution to crystal. And that will help reduce this unfavorable term. So positive terms are unfavorable. That will help us reduce this term, and it will make the process more thermodynamically favorable. Now, another way that can be done is that we can increase the concentration of protein to favor crystallization. So this equilibrium that is there, if we can increase the concentration of this side of the equilibrium, it will be pushed to the right. And that can be done in different ways. One is basically you put the dilute protein solution in a tube like this, and then the tube is inverted and it is put in a centrifuge. So what this will do is that the water is being removed. So this is a kind of centrifuge which allows the removal of solvent. So suppose you have this large amount of dilute protein solution. When you put it in the invert the tube and put it in this centrifuge, the extra solvent will be removed and you will get a very high concentration of protein. So the concentration on the left-hand side will be increased, which will push the equilibrium to the right-hand side. There are other ways of doing this, like instead of removing this, you can remove the so you can have the solvent in a flask like this and then apply vacuum to remove the excess solvent and concentrate the protein solution. Similarly, this is another way. Again, vacuum is applied so that so this is vacuum where you have the dilute protein solution, and using vacuum you are removing solvent to concentrate the protein solution. This is kind of the opposite. You have the dilute protein solution and you're applying pressure. So pressure is being applied, and there is a membrane at the bottom of this tube which allows solvent to pass but will not allow protein to pass. So as you are increasing the pressure that you're applying, more and more of the solvent will be pushed outside the solution. So the solvent is decreasing, meaning that the protein concentration is increasing. Or another way this can be done is called dialysis. So you have this is called a dialysis bag, and it has a semi-permeable membrane. So inside the dialysis bag, there is a concentrated solution of the protein. So and so this red molecule is the protein, and you can see that inside the dialysis bag, if you take the protein along with a high molecular weight molecule like polyethylene glycol, and then you allow it to equilibrate, then again, like we saw in crystallization how we can do. So we saw this method in crystallization, right, with the semi-permeable membrane or the dialysis bag. So this is the similar concept where you can use the dialysis bag to reach the optimum concentration of the protein that is required for crystallization. So these are some ways in which you can increase the protein in the solution so that the equilibrium is pushed towards the right, that is, the crystals are formed easier. Or another way is to add a precipitating agent. So I discussed that precipitating agents are molecules that are not part of the crystal, but they will help the crystal to form. So there are certain molecules called polyethylene glycol. And suppose you have, so this is a protein, and these are the polyethylene glycol molecules. The polyethylene glycol molecules kind of form a coat around the protein, and they help the protein crystallize. But for this, the length of the polyethylene glycol is important. So suppose polyethylene glycol 400 means that 400 molecule. So this is a polymer, right? So if the polymer is of 400 molecules, you can see that the size is too small to act as a precipitating agent. And if the polymer is of 20,000 molecules, then the size is too large. But when the polyethylene glycol polymer is around 4,000 to 8,000 molecules long, then it is of the perfect size to act as a precipitating agent. It forms a kind of layer around the protein and helps the protein crystal to form. And vapor diffusion, we already discussed, the hanging drop or the sitting drop method. Vapor diffusion is also a method to allow the crystallization of the protein. So this is again talking about dialysis, which we have discussed. Or so dialysis, this is the conventional method of dialysis, where you have a semi-permeable membrane in a solution. But now with advancing technology, there are things called dialysis cassettes, and they increase the speed of dialysis by a lot. It is, the concept is the same, but they are kind of, the design helps to maximize the surface area to volume ratio, which speeds up the process of crystallization.
Ma'am, can you please explain the dialysis
Sorry?
once more? Ma'am, can you please
So
explain?
so I'll go back to the original slide. Yeah, so in dialysis, what was happening? You have a very high concentration of protein, precipitant, and solvent
Ma'am,
inside
not
this.
vapor diffusion.
No, this
Ma'am,
last one is
dialysis,
the
that back
dialysis.
one.
See, microdialysis. Vapor diffusion is the
Okay.
first two. The last one is the dialysis. I'm just
Ma'am,
explaining.
hmm.
Continue what you were saying.
Ma'am, actually that pocket one dialysis was there in that diagram.
Which one?
Ma'am, this one, ma'am, this one. Right side
So
dialysis.
So basically, here the large red molecules are the proteins, and the small blue molecules are the precipitating agent. So initially the precipitating agent concentration is too high, and the dialysis membrane is a semi-permeable membrane, so it allows the diffusion of the precipitating agent, but it will not allow the protein to diffuse because it is a large molecule. So here what in vapor diffusion, what you do is you increase the concentration of protein till the crystal starts forming. Here you decrease the concentration of the precipitating agent till that reaches a optimum concentration for the crystal to form. So the precipitating agent is slowly diffusing out into the buffer solution, and inside the dialysis bag, when you are reaching the optimum concentration of precipitating agent, then you can, so we had this, so precipitant concentration is also important for crystallization, right? There has to be a balance between protein and precipitant concentration. So you are adjust slowly as the dialysis is happening, the precipitating agent is diffusing out, and at one point you will reach an optimum concentration of protein and precipitating agent, so that crystallization will begin inside the dialysis membrane. Is that okay?
Yes, ma'am.
Just one second.
Ma'am, so the protein will be crystallized inside the bag itself.
Yes, yes, because the protein cannot escape the bag. The protein molecule is too big, and the semi-permeable membrane will not allow the protein to pass through it.
So the other molecules, what they will do? Is there any
Uh,
advantage?
no, it is just to, so basically, what we had discussed is that if the concentration is slowly changed, then the crystallization will happen properly, perfectly. So the semi-permeable membrane, it is just allowing the concentration to change very slowly so that the crystallization can happen. After the crystallization is happening, again, all the semi-permeable precipitating agent in the outside can be gathered and used for another crystallization. So they can be reused, is what I mean.
That is called dialysate.
Dialysis is just the this diffusion that is happening from inside the bag to outside.
No, ma'am, dialysate. It's written in red.
Dialysate, yeah, 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 dialysate, yeah.
Okay.
So there are some other techniques. So one technique is called this free interface diffusion, where you take in a tube, you take the protein solution in one part and you take the precipitant solution in one part, and you allow them to diffuse into each other. Eventually, when optimum concentration is reached, the crystal will start to form. You can ask that why in protein solution you can't just pour the solution of the precipitant. That is why we, because we want the diffusion, the concentration to change very slowly. So that is why it is put in this kind of a tube, and very slowly the diffusion is happening, and eventually the entire tube will have the same concentration, the throughout the same concentration of protein and precipitant. And when it is, if it is like the optimum concentration, then crystals will form. So this is called free interface diffusion. Or you can have something called a micro batch crystallization. So in micro batch crystallization, very small volume of protein droplet, as little as 1 microliter, is poured into oil. Oil is used because if you just pour that smaller drop in air, it will evaporate. The solvent will evaporate. So it is poured in oil, and so mainly for this, uh, the so you are pouring it in the oil as a drop, and so you will form so this is done to order to get very small crystal samples. So suppose you have the optimum concentration for crystallization, but if you are doing the crystallization in a big tube like this, large crystals can form. But for some particular experiment you want a very small crystal, so you will take one drop of this solution out, and if you just leave the drop in air, what will happen is the crystal will form, but it won't be very nicely ordered, because if you have just 1 microliter of this solvent, evaporation will happen so fast that the crystal will be irregular. So instead, one drop is taken from this tube and it is put into oil, so that in the oil and the generally aqueous solution is used. So oil and water won't mix. So what will happen is that you will have one droplet of this solution in the oil, and then in that droplet, one tiny crystal will form. And so this method is basically used to get very small crystals. Okay. And then the most advanced method that is used is something called droplet based microfluidics. So basically here what happens is you have different three different tubes which are meeting at one place. In one tube you are putting, say, protein, in another tube you are putting the precipitant, in third tube you are putting the solvent. So three different things are coming together to form drops. And so you can at the rate, the concentration of each of the things you can adjust. So suppose in one drop you have 90% concentration of protein, 10% concentration of precipitant. Then in the second drop, you can make 80% concentration of protein, 20% precipitating agent. Like that, in each drop, you can control the concentration of protein, precipitant, precipitating agent, and the buffer solution. So because there are three different tubes and the concentration of each thing can be adjusted, so in each drop you can adjust the concentration and see that where the which is the optimum concentration for formation of the crystal. So you can see the concentration inside the drop is controlled by varying the relative flow rates of the three streams. And this will help you. So instead of carrying out a long experiment, you can vary the concentration in just each drop. So that will give you an idea of the optimum protein concentration. So this is called microfluidics because you're carrying out the experiment in a very microscopic scale. So yeah, this we have already seen, is that this chart we have seen. And so yeah, this is not that. So if there is no nucleation, you can have so this is just explaining the different zones against the crystal growth zone and the nucleation zone. This is not that important. Yes, so back to microfluidics, you can see this is a better illustration. So you have three channels, one which has the protein, one which has the salt or the precipitating agent, and the buffer. And you are creating different drops with varying concentration of the different things, and you are allowing the crystallization to happen. So like that, you can find out that at what concentration the optimum crystallization will be happening. One thing was seen is that the nucleation was sensitive to the flow rate and mixing. So when the flow rate was very low, the nucleation happened very fast, and so crystals were also formed fast. But if the flow rate is high, there was no nucleation visible, and like you can see here, the example is shown. If you have a slow flow rate of 1.85 millimeter per second, many small crystals will be formed because the nucleation is favored. Uh, and in the fast flow rate, number of nucleation is very less. So you will have a few large crystals. I'll just repeat this once more. What was seen from this experiment is if the flow rate is slow, you have multiple nucleations happening everywhere. So if there are multiple nucleations, you will have many small crystals. But if the flow rate is fast, you will have very few nucleations. So few nucleations means you will have few large crystals. So depending on your application, you can get small crystals or large crystals depending on whatever is your application.
Um, uh, the that protein 30 milligram per ml and buffer 0.1 molar.
Mhm.
What is this saying? Means the left hand side diagram.
Yeah. So see, from this initial experiment, the optimum concentration of crystallized, so you can vary the protein concentration, vary the buffer concentration, and vary the salt concentration. Salt is your precipitating agent. By varying these concentrations in the initial experiment, it was found that at 30 mg per ml protein, 0.1 molar buffer, and 2 molar salt, your optimum crystallization condition is achieved. Now you can control the size of crystal, whether you want many small crystals or you want a few large crystals, and that can be controlled by controlling the flow rate. So basically this is the optimum concentration that was found for crystallization to happen.
And ma'am, capillary is that tube, na?
Yes, it is just this tube through which the drops are passing.
And ma'am, the interfaces are where the protein buffer and salt meet.
Yes, yes, yes.
Okay, ma'am.
Yeah. So you can see this is again showing the like different kind of what we just discussed, that you have different input channels. You're including protein, salt, buffer. In some cases you might need a second precipitating agent like polyethylene glycol, and you can find out the optimum range of each of these, so you can optimize the concentration. uh, you can optimize the salt. So different ways. What we just talked about basically is that you can optimize each of these different things. So yeah. Now is the differentiating between protein and salt crystals. So there are certain dyes which will only stain proteins and they won't stain salts. So that is one way in which you can differentiate whether it's a protein or a salt crystal. Another thing we discussed is that protein crystals have a large amount of water, that is water or other solvent. That is why if you crush a protein crystal, it will very easily crush into fine pieces. But the salt crystal is difficult to crush. Then, if you allow dehydration, protein crystal is stabilized by a large amount of solvent. So, if you allow the solvent to dehydrate, the protein crystal will lose its shape, but the salt will not. And finally, they have very characteristic X-ray diffraction pattern. So this kind of a diffraction pattern is found in protein crystals, where you have these small dots arranged all over. But the salt diffraction pattern, as you can see, that is why X-ray crystallography is not generally used for salts, because it is not a you can see this pattern is there are a lot of white spaces. It's not a very regular well-defined pattern. So diffraction patterns, these are just some sample diffraction patterns. So if you can have a question asking, giving you this diffraction pattern and asking whether it is the diffraction pattern of a protein or a salt. So it's a very characteristic pattern, so you can say this is a salt. And if there is this kind of a black ring and well defined dots all over, that is a very characteristic of a protein diffraction pattern. So these are some qualitative differences between your protein and salt crystals.
So salt
And
crystal, what
now,
will be the difference? Salt
huh,
crystal, what will be the difference in X-ray diffraction pattern?
so it you can see just you can look at these two patterns. No, in salt you can see the number of dots are much lower, and there is a lot more white space. So you just compare this image and this image. The one on
Okay.
the right is your salt crystal, one on the left is your protein crystal.
I
So
thought right one is nothing. It's
oh
just
no, no,
the.
I understand. That is what salt is almost like nothing only. That is why it's not used. So you can
Okay,
see
ma'am.
salt crystal right. So yeah. And there is a kind of mechanism that is used. This is like the instrumentation. So you have the crystal in the solvent. A loop is kind of, this is called a loop. It picks up the crystal, and so you have the crystal fixed here in the loop. uh, then it is put on this instrument called goniometer, which we'll just discuss. So, loop is necessary to pick up a crystal across the surface of the drop and avoid unwanted dehydration before freezing. So, basically, the first step is to pick up the crystal, and we want to pick up the crystal along with the solvent, because if we um, pick up the crystal without the solvent, the crystal can lose its shape. So then this kind of a nylon loop is used to pick up the crystal. Then the crystal is flash frozen. Flash frozen means that very fast, rapid freezing to minus 70 degrees Celsius is done. And why does it have to be very rapid and very low temperature? Because if you just freeze at say minus 10 degrees Celsius and you do a slow freezing, the ice will form in this kind of a hexagonal orientation. You can see 6 water molecules attached by hydrogen bonds. This kind of orientation ice will form, and there will be a lot of empty space between the ice. This is not wanted. What we want is that in the protein, however the water molecules are placed in the protein, we want the water molecules to freeze in that same orientation. We don't want the water molecules to reorient and form this hexagonal ice. That is why the crystal, after taking in the loop, is immediately dipped in liquid propane or freon or nitrogen at minus 70 degrees Celsius. What this will do is that whatever is the orientation of the water molecules in the protein, it will immediately freeze the water molecules in the same orientation. So the 3D structure of the protein is maintained. But if you slowly, if you put it in, say, just a normal refrigerator where the temperature is minus 5 degrees Celsius, then the freezing will happen very slowly, and that will allow the water to reorient and form this kind of hexagonal ice-like structure. So this will completely destroy the structure of the protein. So that is why this flash freezing step is done. And another thing is that freezing, what it will do, X-ray, we know wavelength is very small, and wavelength small means high energy. So when you're passing high energy X-ray through the crystal, it can damage the crystal. So the freezing of the crystal in a cool temperature protects it from damage during the data collection process. Now, after freezing, it is mounted on this instrument called a goniometer. So there are goniometers are used for measuring angles between different crystal phases. Once the specimen has been selected and put on the loop, it is mounted. So you can see this line. This is the loop and the crystal is on the loop. Now this goniometer is oriented at a certain axis, the phi axis, and there is an angle between, so this the phi, instead of the normal XYZ axis, a new axis system is defined where the axis which passes through the goniometer is the phi axis, and the axis 90 degree to that is the omega axis. So from this plane, the angle that the goniometer is forming, that is called your magic angle, or which it has been optimized. Again, we will see why this value, why we get this value in the next class. For now, I'm just saying that it has been optimized that if the goniometer is placed at an angle of 54.74 degrees with respect to this plane, then we get the best resolution of the crystal. So basically, first using the loop, the crystal is picked up from the solution. Then it is flash frozen by dipping into liquid propane or freon or nitrogen at minus 70 degrees Celsius. And then it is mounted on the head of the goniometer, and the goniometer is oriented at an angle of 54.74 degrees called the magic angle, at which the X-ray resolution is best. So that is the instrumentation of this process. And
means on the goniometer that the sample is planted.
yes, the loop containing the sample is planted here.
Ma'am, with the loop only it is planted, or just the sample it is planted?
I have to check actually. I'm also not. I thought it is with the loop, but I'll just check once. No, actually the loop is just for picking it up.
Loop.
Actually, it is
Yes.
loop is just for picking it up. No, without the loop only. Sample is just planted on the top.
And because if we will put the loop, then how will that image will be formed?
Correct. Yeah, correct. You're correct. So after crystallization, this is the most difficult process because you have to get a perfect crystal structure. Then the X-rays are passed to get a diffraction pattern. Basically, when the X-rays are passed through the crystal, wherever the X-ray is absorbed by the atoms in the crystal, you will get these dark spots. And depending on whether the electron density is high, then the density of these spots will be high. If somewhere electron density is low, density of spots will be low. So using this pattern, we can create an electron density map. And from the electron density map, the atomic model can be predicted. These are mainly done using softwares which will fit a particular electron density to a particular functional group. So these steps are done by softwares. So the diffraction pattern is basically what helps in reconstructing the three-dimensional structures. We will go into the details of the steps later. It is just this basic process that is happening. And finally, I'll just quickly discuss how the X-rays are produced. So basically, you have a filament here, tungsten filament, which is heated. When it is heated, electrons are emitted from this filament, and these electrons are called thermionic emission because it is emitted due to heat, and it is accelerated towards a anode. So you have a positive charge here. The electrons are accelerated towards the positive charge, and it hits the anode, which is generally made of tungsten. When the electrons hit the anode, then the X-rays are produced. And how these are produced in an atomic level? So this electron is the one that is produced from the cathode by thermionic emission. When the electron hits the material of the anode, what it does is that it will knock out an electron from the atom of the anode. So this electron was in the K shell of the anode. Because of being hit by the thermionic electron, it is knocked out. So there is a vacancy here. In order to fill up this vacancy, one electron from an outer shell will jump to the inner shell, and the energy released in this process is basically what forms the X-ray. So this is your the atomic origin of X-rays. So this was the last slide that I had. I will now discuss some sample questions. So if you have any doubts here, please ask. Okay, I'll move on to the sample questions. These are basically like your assignment questions only. So one question is asking that what information about a protein structure can be obtained from X-ray crystallography?
3D structure.
Yeah. So because the crystal is fixed, so you cannot know anything about dynamics or folding mechanism. NMR happens in a solvent state. So NMR can give you idea about dynamics and folding mechanism, but X-ray will only give you idea about the 3D structure. Then it is asking that which of the following is not true for a protein crystal?
Option D, protein remains
Yeah.
non-functional.
Yes. So I already discussed that because the protein is crystallizing along with solvent molecules, the protein is flexible even in the crystal. So it can, so this is a protein crystal, and you can, which is an enzyme, and you can add the substrate in the crystal and the product will be formed. So even in the crystal, it remains functional. That is why option D is wrong. Then this is asking nickel NTA affinity chromatography is based on what?
Option A, nickel with histidine tag.
Yes, that is a very straightforward question. We have already discussed. So any affinity chromatography is based on very specific interactions. Sorry. Have somehow already. Uh, which of the following amino acid side chains has 3 different pKa? So this is kind of actually based on the previous classes thing, not this classes. So it is histidine. So histidine molecule has a basic side chain. So initially in very acidic conditions, the COH is protonated, the main chain amine is protonated, and the side chain imidazole is also protonated. Then as the pH becomes more basic, first this hydrogen is lost. The COH becomes CO minus. After that, the side chain hydrogen is lost. This NH plus in the ring becomes NH. And finally, the main chain amine hydrogen is lost. From NH3 plus, it becomes NH. So because it has three hydrogens it can lose, it has three different pKa. This is kind of related to the previous class, not this class, but still, yeah. So next question is asking that which of the following statements is true when performing size exclusion chromatography?
Ma'am, option D, smaller protein elute last.
Yeah. So as you can see, the small, I discussed also, small proteins fit into the pores and the large proteins will elute out first. And then as you add more solvent, the small protein is eluted out last. This is also very simple, that which of these is
Loop.
used to, yeah. So we have already discussed that the loop is used. Uh, this is asking that if we look at the thermodynamic equation of crystal growth, which of the following statements is false about the thermodynamics of crystal formation?
Ma'am, is it entropy of solvent increases?
No, think about it, that see, solvent entropy will it is asking which statement is false, right? So uh, thermodynamically unfavorable is false because we saw that the net delta G is negative. So 1 second. So net delta G negative, so it is modestly favorable. This is true. Unfavorable is false because unfavorable means delta G is positive. Entropy of protein decreases is true because protein, that they were single, like there were many protein molecules and there is one protein crystal. So entropy of protein decreases. Entropy of solvent increases is also true because if you go back to that thermodynamic slide, so you can see in this slide, initially solvent was trapped here, but after crystallization, solvent becomes free, right? So entropy of solvent increases upon crystallization. So
Ma'am,
your
how, ma'am, solvent, entropy of solvent will not, means there will no change in the entropy of solvent.
see, initially 100% of your initially the solvent is uh, solvating each of the protein molecules separately. So the solvent is attached to the protein molecule and they are not free to move. So they have low entropy. After protein is forming the crystal, they are, so these horizontal ones are the proteins. They are coming together to form the crystal. Then the solvent will solvate the whole crystal. They are not individually solvating. So many of the solvent molecules are now released, right? They are not trapped with the protein. So the solvent molecules are free from many, some solvent molecules will still be solvating the protein. Say if we have 100 solvent molecules, and each protein is being solvated by 5 solvent molecules. But when the protein crystal is formed, some of the solvent molecules will be solvating the protein crystal, but most of the other solvent molecules will be released. They will form a part of the bulk solvent. So they will have higher entropy.
Okay, ma'am.
This is already answered this anyway. So which of the terms contribute to enthalpy? Yeah. Which of the following terms contributes to enthalpic stabilization when the crystal is being formed? So the answer is hydrogen bond. So when you have a crystal, second. When you have a crystal, you can have hydrogen bonds inside the crystal, and you can have lattice contacts. Like the crystal is stabilized by hydrogen. So suppose you have one protein molecule, another protein molecule in a crystal, there can be hydrogen bonding between them, which is stabilizing the structure of the crystal. There can also be van der Waals interaction and everything. So these contribute to the enthalpy. Degrees of freedom contributes to entropic terms. So it will contribute to S, not H. Solubility doesn't really contribute to any of these. So the correct answer is only A, that is hydrogen bond. This degrees of freedom is contributing to entropy, so it is not part of the answer.
Solubility can also contribute to entropy.
Oh yes, solubility can contribute to entropy. You are correct. Very correct. True. But it will not contribute to enthalpy, so it is not part of this answer.
Okay.
So which of these statements are true? So the answer is both A and B, because we
Both and
discussed
B.
that crystals must be flash frozen to minus 70 degrees Celsius before X-rays are passed through it in order to protect the crystal. And this is done by dipping it in liquid propane or freon or nitrogen. So you can see one difference also. If there is flash freezing, then the whatever orientation the water molecules in, they will freeze in that orientation. So the structure of the crystal is maintained. But if there is slow freezing, then the water molecules can form that hexagonal ice-like structure I discussed, and the structure of the crystal will get broken down. So that is why this flash freezing is important. The last question for today is that in which of the zone the protein crystals are soluble? So this is your under saturation zone. See, nucleating zone, the crystal is coming out, no? So it actually, the question is wrong. It should not say protein crystals are soluble. It should say protein is soluble. So just ignore the word crystals. I'll correct this question later. So in which of the zone protein is soluble? It is your under saturation zone. In the nucleation zone, crystal is forming, so it is no longer soluble. So yeah, this question itself was misleading. Okay.
Ma'am, can you explain the graph once again? Means
Yeah.
what is nucleation zone and
yeah,
why
so
it is called nucleation zone?
Yeah, so in the soluble state, so there is first of all an equilibrium between the white region and the light blue region. In the white region, the protein is in solution. There is no crystal forming. That is the under saturation zone. In the white region, it is called the crystal growth zone. So crystal is forming, but it is again dissociating. That is why in this light blue region, we call it transient crystals. So crystal is forming, dissociating. This equilibrium is existing. But once it passes into the nucleation zone, it is irreversible. So once it reaches the dark blue nucleation zone, say one small crystal has formed, and as the concentration is maintained around that small crystals, more crystals will accumulate to form a larger crystal. So this is the crystal, that is why it's showing crystal growth. So once a nucleus is formed, immediately the crystal will grow. But if you increase the concentration beyond that into the precipitation zone, then it will like the concentration is too high. At that time, the crystal will just crystal will not form. The protein will precipitate out in an so in precipitation zone, no, you will just have loose, like the protein molecules will just precipitate out individually. There will be no formation of crystals. So that is the amorphous state. So this is the crystalline state, this is the amorphous state. In the precipitation zone, it will precipitate out in the amorphous state. So that is why if you are within this range, the crystals are not stable. But if you reach the nucleation zone, it is the best region, best concentration, both in terms of protein and in terms of precipitant, best optimum concentration of both to get the best growth of the crystals.
Ma'am, so here it is said about proteins are soluble. Means protein are soluble in the solvent or
In the solvent. So when it is in this white region, no, proteins are soluble in the solvent. As you are saturating the solution, you know that after a point, the solution is so saturated that it is saturated, then it will start crystallizing out. So in which zone protein is soluble is this white region, where the concentration of protein is low enough that crystal is not forming. That is, it is called the under saturation zone. And the rest of this entire zone, which includes this light blue, dark blue, red, it is called the supersaturation zone, where the solution is completely saturated, and after that the crystals start to form.
okay, ma'am. And crystal growth zone is the that light blue zone.
Equilibrium, where crystal is forming but it is again dissociating, so it is not a very stable zone.
So why it is said crystal growth zone if it is not?
It's not just the terminology. I'm not sure. It should not be called technically, but it is just called that. Maybe because it starts growing. I don't know.
And in the nucleation zone, it is fully formed.
Yes. In the nucleation zone, nucleus starts forming, and once a nucleus starts forming, it is irreversible. So this arrow is only one direction. Like if you enter the nucleation zone, now the crystallization will continue, and around the small nucleus, larger and larger crystals will start to form.
And then finally it will get precipitated and come out.
Uh,
Okay, ma'am.
okay. So that is all for now. I'll share the video as well as the slides. And do you have my email address? I gave it in the previous classes. So if you want to, if you have any doubts regarding this lecture or the next week's lecture, you can just email me with your doubts in your email address. So I'll start the next session by clearing the doubts. Do you have my email address or will I give it?
No, ma'am, I don't have.
I'll just write it. You can note this down. And you can mail me your doubts before next Monday, so that in next Monday's class, I'll first start by clearing the doubts and then I'll discuss the next week's lectures.
Okay, ma'am.
Okay. Thank you. I'll end the
Thank you.
class here. Sure.
Thank you so much, ma'am.
Okay, sure.